For the past couple of weeks, we've been bogged down with looking at chemistry and molecules, but that was all a setup for what we finally get to do starting right now, which is to take a look at cells, and that's going to be the focus for the rest of the quarter. So in the assignment that you hopefully did before looking at this video, you took a look at the typical structure of cells and you took a look at the way that cells can be categorized. So what we're going to do here is follow up on that information by taking a look at these things in particular. So you saw that there is a category of cells. So in the knowledge check assignment before this video, you saw that there is a category of cells that contain these things called organelles inside of them. And so in the knowledge check assignment, we took a look at what some of those organelles did. And so a lot of them do some pretty different things. And so it's easy to get the impression that these organelles work independently of one another. But the reality is that a lot of organelles actually work together. And so we'll show some examples of how organelles work together in this video. After that, we'll focus on one particular organelle, the one called the lysosome. And we'll talk about how the lysosome works and what the function of the lysosome is. And then at the end of this video, we'll take a look at a component of cells called actin and what it does and why it's important to cells. So first we'll start here. We'll take a look at some examples of how different organelles work together. But before looking at that, what if we do this? So hopefully you remember from the knowledge check assignment that one way of categorizing all of the cells out there is based on whether they are a eukaryotic cell or a prokaryotic cell. And so hopefully you can take a look at these two drawings of cells on the screen and pick out which one represents the eukaryotic cell and which one represents the prokaryotic cell. So it turns out that this big guy on the left represents the eukaryotic cell and the smaller guy on the right represents the prokaryotic cell. And there are a few things that are kind of the giveaways that this is a eukaryotic cell. First, and the thing that defines eukaryotic cells is the fact that its DNA is enclosed within a nucleus. So eukaryotic cells have a nucleus, and prokaryotic cells, again, they have DNA, but it's not enclosed within a nucleus. It's just kind of free-floating within the cell's cytoplasm. Another thing is that eukaryotic cells have a lot more organelles inside of them. So the nucleus is just one example of an organelle. All of these other things are organelles as well. Basically what an organelle is, it's a structure inside of a cell that itself is surrounded by a membrane or is made of a membrane. And so all of these things are membrane structures. Prokaryotic cells tend not to have any of that stuff, or at least not as much membranes inside of it. It just has the membrane that surrounds the entire cell, but it has no organelles inside of it. And then the last thing that might be a giveaway that this is a eukaryotic cell is that it's freaking huge compared to the prokaryotic cells. So eukaryotic cells are typically bigger than prokaryotic cells. Again, the thing that defines a eukaryotic cell is that it has a nucleus that surrounds its DNA. Basically, the function of that nucleus is to protect that valuable DNA molecule that's inside of it. And so if you consider that the function of this nucleus is to protect the DNA inside of it, it might surprise you to know that the nucleus actually has holes in it. And so you can kind of see them here, these little pores. Here's another look at it. So here's a picture of just the nucleus. It's been isolated from the rest of the cell. Here's the nucleus. Here's one of the organelles that it's associated with. And it's actually labeled here that there are pores inside of the nucleus. Here's an even bigger drawing of it, an even a bigger close-up of it, that there's a pore that you find in the nucleus. Here's an actual picture of a nucleus, and this is the nucleus of a rat liver cell. And you see that there's actually a hole there in the nuclear envelope. So you might be thinking, what gives? Right? So if the function of this thing is to protect the DNA inside of it, why in the blue heck would it have holes? It seems like holes would make the structure weaker. Wouldn't you want the structure to be as strong as possible so that it can do the best job possible at protecting the DNA inside of it? Well, it turns out that these holes have a pretty important function. There's a reason why they're there. And basically they're there in order to allow the DNA that's inside of it to work with other organelles that are outside of the nucleus. So here's what I'm talking about. Here's a cartoony drawing of a cell. This represents the nucleus of the cell. And these things outside, so there's a lot of things outside of the nucleus, so we're just focusing in on one of those things. These things here represent ribosomes. So keep in mind that this is not drawn to scale, so ribosomes typically aren't this huge compared to the nucleus, but since we're focusing in on the ribosomes, I thought it might be okay to make them bigger than they actually are. Again, what you find inside of the nucleus of a eukaryotic cell is DNA. We mentioned in the knowledge check assignment that the function of DNA is that it encodes for proteins. In other words, DNA has the information on it that tells the cell how to make different kinds of proteins. You might remember that where the proteins are actually made are at these places, are at the ribosomes. So there's kind of a disconnect there, right? So the place where the proteins are actually produced are out here in the ribosomes, but the instructions that tell the cell how to make the protein is locked up in here. Wouldn't you want those things to be in the same spot? So if you're making a cake, right, you want the oven and the recipe all to be together in the kitchen in order for you to make the cake. You can't have the recipe like in a different city because you can't read it and do the actual cooking at the same time. So the same thing here, if something gives, there has to be a way to get the instructions together with the place that the proteins are actually being made in order for that all to happen. And so that's where the holes come in. So we mentioned that the nucleus contains pores or holes, and basically they're there to allow the information for making the protein to get together with the place where the protein is actually produced. Here's the thing though, DNA is a really important molecule. And so in eukaryotic cells, the DNA stays inside the nucleus all the time so that it stays pretty protected. So what happens is that it's not the DNA itself that goes out to the ribosome, but instead, kind of a cheap knockoff copy of the DNA is produced, a temporary molecule that can leave the nucleus. And so basically this molecule here, we'll learn later that it's called messenger RNA, but for now let's just call it a cheap knockoff of DNA. It's a temporary, it's a molecule that acts as a temporary holder of genetic information that tells the cell how to make a protein. So it's the one that travels out of the nucleus over to the ribosome so that the cell can read that information and know how to make a protein. And again, this is really important because proteins do many different things for a cell. So some of them act as enzymes that allow different chemical reactions to occur. Some of them act as structural support. Some of them are antibodies that protect us against bacteria and viruses. Proteins just do many different things that allow a cell to stay alive. But the point that we're making here is that different parts of a cell work together. That's the reason why you need holes or pores in the nucleus. So why does the nuclear envelope have pores? It's because genetic information, it allows the genetic information to go to the ribosomes so that proteins can be produced. All right, so kind of going back now to the main function of this nuclear envelope, which is to protect the DNA inside of it. Sure, there's holes in it, but it still does a pretty good job at protecting the DNA inside of it. So again, if you know that if you consider the function of the nucleus, what might you guess would happen if the nucleus were somehow to become degraded? So what if the structure of the nucleus were to become weakened? What do you think might happen then? Well, you might have guessed that basically what occurs is that if the nucleus is weak, that means that the DNA inside of it is less protected. So, for example, enzymes and other things might be able to seep in and start degrading that DNA. Whenever someone's DNA becomes degraded, one of the results of that is that you start seeing aging occur. So it turns out that there is a condition where that's the case, where so if you take a look at the nucleus inside of a person, or the nucleus inside of the cells that make up a person, that nucleus is very weak in terms of the structure. And what that results in is a condition called progeria. And one of the hallmarks, or the hallmark of progeria, is accelerated aging. So this is a story that was in National Geographic not too long ago, and this is someone who was affected by, who had progeria. Like the caption says here, Sam Burns, he, in this picture was only 17 years old, but you see that he looks way older than 17. And again, it's because his cells have started breaking down prematurely. The reason why they were breaking down is because if you took a look at the nuclear envelopes inside of all of his cells, they were very prone to being damaged. Specifically, here's the reason why. You might remember from the knowledge check assignment that if you take a look at, here's just the nucleus now, so the cell that owns this nucleus would be out here. It's just not being shown. This is just the nucleus of the cell. If you consider the nucleus, you might remember that there are proteins that help to maintain the structure of that nucleus. One of those proteins is called the lamina. And so basically what happens if someone has progeria is that the lamina of the nucleus is misformed. And so those with progeria can't make a properly formed lamina, which means that the nucleus is more weak, which means that the DNA inside of it is more prone to breaking down, and that's what then causes this accelerated aging to occur. So again, the point of this slide is that the nucleus, it does serve a protective function. It protects the DNA that's inside of it, and here's kind of the proof of that. So if you have a condition where the nucleus is weak, then you see the effects of the DNA being broken down prematurely. So, all right, so we've mentioned that in order for proteins to be made, the DNA has to work together with the ribosomes in order for that to happen. What we'll do next is say a little bit more about proteins and mention some of the other parts of the cell that can be involved when proteins are produced. So again, it's kind of worth mentioning all this because proteins are so important to cells. They do many different important jobs for a cell. We mentioned that ribosomes are the place inside of the cell where the proteins are actually produced. You might remember from the knowledge check assignment that there are different places where you find the ribosome. So generally ribosomes are found outside of the nucleus, but there are two main places outside of the nucleus where those ribosomes are found. Some of those ribosomes are stuck to this structure called the endoplasmic reticulum. And so whenever you consider rough endoplasmic reticulum, that's what makes it rough, is that it has ribosomes attached to it. Ribosomes, though, are also found out here just free-floating in the cytoplasm. And so one of the differences between the ribosomes that are out here in the cytoplasm and those that are stuck to the endoplasmic reticulum is the type of protein that they end up producing. So here's what I mean by that. Here's a cartoony version of a cell, cartoony drawing of a cell. So again, here is the rough endoplasmic reticulum that has ribosomes studded on it, and there are also ribosomes out free-floating in the cytoplasm of the cell. What makes both of these ribosomes similar is that they both make proteins. So we'll show this guy making a protein, the ribosome that's out in the cytoplasm. The ribosomes that are part of the rough endoplasmic reticulum also make proteins. But the difference is that the proteins that these ribosomes produce tend to be the type of protein that the cell ends up secreting, ends up spitting out to the outside. Kind of like this. And so there are different reasons why a cell might want to secrete a protein. So there's a type of cell of the immune system called a B cell that makes these things called antibodies that it spits out. And what those antibodies do is that they go bind to bacteria and viruses that might have found their way inside of the body as a way of kind of neutralizing those bacteria and viruses. Another reason why a cell might want to secrete a protein is because some proteins act as communication molecules that affect the activity of other cells. Right, so there's many different examples of why a cell would make a protein and then spit it out, secrete it. The idea here is that the type of ribosome that makes those proteins aren't the ones that are free-floating in the cytoplasm, because those tend to make proteins that stay inside of the cell, maybe to act as an enzyme or maybe to provide structural support for a cell. Whereas those ribosomes that are part of the rough endoplasmic reticulum, they tend to make secretory proteins. So in other words, proteins that end up being secreted or spit out of the cell. It turns out that there can be actually yet another organelle that can be involved in making proteins, especially those that end up leaving the cell. So we'll add another organelle to the mix here when it comes to the act of making proteins. So if you go back to this picture here, we've taken a look at the DNA inside of the nucleus. We've taken a look at the rough endoplasmic reticulum. It has ribosomes on it for making proteins. This structure right here, you might remember from the knowledge check assignment, is something called the Golgi apparatus. And you might recognize, or you might think to yourself, look, it looks like this structure, the Golgi apparatus, is very similar looking to the rough endoplasmic reticulum. It just looks like a bunch of folded membranes. And so one of the kind of adages in cell biology or biology in general is that structure relates to function. So in other words, if an organelle has a particular structure, if it has a particular look, it's because it allows it to do whatever its job is, whatever its function is. So if you have this structure here called the Golgi apparatus that has a very similar structure to the rough endoplasmic reticulum, then you might deduce that its job or its function is also very similar to the rough endoplasmic reticulum, and that actually turns out to be the case. So you know that the rough endoplasmic reticulum is involved in making proteins. Turns out that the Golgi apparatus is also involved in making proteins. These two organelles actually work together in order to produce largely proteins that end up being secreted from the cell. And so here's what I'm talking about. We'll kind of redraw this cartoony style. So everything in that yellow line is going to be redrawn to look like this. So this blue thing represents the rough endoplasmic reticulum. This represents the Golgi apparatus. These represent the ribosomes that are attached to the rough endoplasmic reticulum. We're saying that these two things work together in order to make proteins that end up leaving the cell. The way that works is that, again, it's at the ribosomes where proteins are produced, at least initially. So there's a protein being produced. Once it's produced, so if you're talking about a ribosome that is producing a protein and that ribosome is part of the rough endoplasmic reticulum, what ends up happening in order for this to work together with that is that the protein ends up budding out of that rough endoplasmic reticulum. And that looks something like this. It buds out. And so when that occurs, basically now you have a freshly made protein that's floating around inside of the cell, inside of its own membrane. And so whenever you have this structure here, basically a membrane that has a protein inside of it, it's called a vesicle. So again, vesicle is a membrane structure that stores substances. That's a more general definition of a vesicle. Here, the specific substance that's being stored inside of it, at least temporarily, is a protein that was produced by the ribosome. What can occur with that vesicle is that it can actually fuse together with this Golgi apparatus, kind of like this. So that's the way that proteins end up being shuttled back and forth, or actually this is more of a one-way thing. That's the way that proteins end up being transported from the rough ER over to the Golgi apparatus. And one of the reasons why the rough ER would pass a protein along to the Golgi apparatus is so that the Golgi apparatus can make some modifications to it. And so an example of a modification that the Golgi apparatus can do to a freshly made protein is to add carbohydrate molecules to it. And so there are some proteins that in order for them to function properly have to have carbohydrates on them. We'll take a look at some examples later on in the quarter. But basically that's what the Golgi does, is that it adds some things that will make the protein fully functional. A lot of times this protein that is produced as a result of the rough ER and the Golgi apparatus working together is a protein that ends up being secreted or spit out of the cell. So the way that happens is that this protein ends up budding out of the Golgi apparatus. So we just added another carbohydrate there, but it ends up budding out of the Golgi apparatus to form another vesicle. And it's this protein, after it's been completely modified and completely built now, that's the version that will end up leaving the cell. So to sum up what we've just mentioned here, the take-home point is this. We've been taking a look at how the rough ER and the Golgi apparatus work together to make a protein. Here's kind of how all of that works in order. First, the ribosome that's attached to the rough ER makes the protein. That protein then buds off of the rough endoplasmic reticulum. Remember, that forms something called a vesicle that then goes over to the Golgi apparatus where it modifies the protein. For example, it can modify it by adding carbohydrates. Once that protein has been fully modified, it can then bud off of the Golgi apparatus to form this structure here, and then from there, that's when it can leave the cell. So it's secreted from the cell in order to do some other function. And the way that it ends up leaving the cell is like this. So what if we redraw all of this? So here's the nucleus of the cell, here's the rough endoplasmic reticulum, here's its Golgi apparatus, and here is the vesicle that it just produced. In order for this entire cell then to secrete the protein that's inside of that vesicle, what has to happen is that the vesicle fuses together with the outer membrane of the cell, kind of like that. And so once that fusion has occurred, that can then allow that fully modified and complete protein to leave. So one of the ways, again, that proteins can be excreted by a cell is by having the membrane of the vesicle, and this is the vesicle, again, that has the protein inside of it, fusing together with the outer membrane of the cell. Right? So the main point of everything in this video up to this point is that if you take a look at the organelles inside of a eukaryotic cell, a lot of them work together in order to do things. For example, a lot of those things work together in order to produce proteins that can be secreted from the cell. What we'll do next is focus in on two parts of the cell and just mention a little bit more about how it works. So this next thing we're going to take a look at is it's an organelle called the lysosome. And that's what represents the lysosome in this picture. And to kind of tie it together with what we've just talked about, basically what the lysosome is, it's a semi-permanent vesicle that contains proteins whose job it is to break down things. So there are enzymes inside of this lysosome that end up digesting things. Enzymes are a kind of a protein. And so what we'll take a peek at next is how that occurs. So we'll kind of redraw this. So here's a cell, here's its nucleus, and then not drawn to scale, here's the lysosome. So typically the lysosome isn't going to be almost the same size as the nucleus. And inside, again, what you find inside of the lysosome are these digestive enzymes. And so there are several reasons why a cell might be interested in digesting things. So a cell might digest some nutrient molecules. The example that we'll take a look at here is digestion of bacteria. So there are white blood cells whose job it is to eliminate and kill bacteria that might be trying to cause disease. One of the weapons they have for killing bacteria are these lysosomes. And so it's kind of a good idea to have these digestive enzymes enclosed within this membrane to form this lysosome. Because if it wasn't, right, if these digestive enzymes were just floating around willy-nilly inside of the cell, they could start breaking things down inside of the cell itself, and that would not be a good thing. So what we're going to show you here is, well, how are these enzymes unleashed so that they can do things? So again, an example of a cell that makes good use of its lysosome are these white blood cells that have the ability to take in bacteria that they encounter. So that red thing represents a bacterium that might be trying to do the body harm. And so this is the way that it takes it in. Once it's in, the goal is to eventually kill that bacterium. And again, these white blood cells, these immune system cells, can use the enzymes in the lysosome in order to kill it. The way that it does it is that it has the membrane that's surrounding this bacterium fused together with this outer membrane of the lysosome, kind of like this. And so when that occurs, that allows these digestive enzymes access to the bacterium that just got taken in. And so once they have access to that bacterial cell, those enzymes can do their thing, which is to break down that bacterial cell. Right? So lysosomes have enzymes contained within them but this is how it kind of unleashes those enzymes. It doesn't completely unleash the enzymes so that the entire cell becomes endangered, but it does so by having by fusing with other things. So so what just happened there? The bacterium just got digested. There it is, pieces now, not a whole bacterium anymore. So again, we're taking a look at how lysosomes digest cellular materials. For example, bacterial cells that might have gotten taken in by a white blood cell. This is how it happens. The material to be digested actually fuses together with the lysosome. Once that fusion happens, that allows the digestive enzymes to break down the material, because those enzymes would then have access to the digested thing. All right, so then the last thing that we'll take a look at in this video is we'll look at a different part of a eukaryotic cell and mention what it does and why its function is important. And so again, here's our, I seem to really like this picture of a cell. So we're going to go back to this picture of a cell. You might remember that one of the parts of a eukaryotic cell is a part called the cytoskeleton. And so the way that you might be able to visualize the cytoskeleton is if you were to take out all of these organelles that are floating around inside of the cell. And if you do that, what you would have left over is the cytoplasm, which is the jelly-like stuff inside of it, and also this meshwork of protein fibers that are just all running throughout the inside of the cell. And so what you see here is a cell that's been stained with something that specifically colors those protein fibers that make up the cell's cytoskeleton. So again, just to remind ourselves, the cytoskeleton is this meshwork of protein fibers. You might remember from the knowledge check assignment that there are actually 3 kinds of protein fibers that make up the cytoskeleton. So if you look really closely, if you could look really closely at these protein fibers, you'll see that there are 3 different sized protein fibers, and they're all given a different name. Right, so when you consider those protein fibers, there are 3 types. The smallest, in terms of diameter, the smallest ones are these things called microfilaments, or actin. Right? So micro means small, so that kind of makes sense that you use this term microfilament to describe the thinnest protein fiber that you find in the cytoskeleton. The medium-sized protein fibers are these things called intermediate filaments, and then the biggest ones are these things called microtubules. So in this case, tubule means just the biggest one that you find within the cytoskeleton. We actually mentioned all of this already in the knowledge check assignment. What we're going to do here is focus in on one of these types of protein fibers, and that's going to be the actin. The reason why I thought it might be worth mentioning what actin does is because I know a lot of you guys are going to go on to anatomy, and one of the things that you'll take a look at in anatomy and also in physiology is muscle cells and muscle movement. And it turns out that this actin that you find in muscle cells plays a big role in allowing for muscle movement. So I just thought I would mention that here so that you'll at least have heard of it before you get to your anatomy class or your zoology class. So actin, again, is involved in moving muscle cells, which then allows limbs to move. Here's how that happens. Here's a cartoony drawing of a muscle cell. These red things here represent the actin protein fibers that you find in muscle cells. Basically, in order for a muscle cell to allow for a limb to move, that muscle cell has to contract. That has to get smaller. And it turns out that these actin protein fibers, again, play a big role in that. The way that they do that is that they interact with another type of protein that will represent by these green lines. And what that protein is called, they're called myosin proteins or myosin filaments. And basically what occurs is that these parts of the myosin filament act kind of like hooks that can grab onto the actin. And then after grabbing on, they kind of walk along each other. That allows this actin microfilament or this actin protein fiber to then kind of pull from the inside of the cell. That allows this actin protein fiber pull on the inside of the cell, and the overall effect is that it allows the cell to contract or get smaller. here's a movie of that happening. So in this picture here, again, the red represents the actin. It's a type of protein fiber that's part of the cytoskeleton. The green represents the myosin. These blue things here basically represent the outside of the cell. If you're wondering, these yellow dots here represent calcium, and calcium is necessary in order for muscle cells to contract. So if calcium is around, basically what that allows for is this. So again, basically those myosin molecules have these hook-like structures that allow them to grab onto the actin, and when it does so, it kind of pulls those actins together. And the consequence of that is that allows the cell to contract. So you see the outsides of the cells here getting closer to one another. So if you have a whole bunch of muscle cells contracting, that allows for limb movement, kind of like this. So when the cells inside the bicep, for example, all contract, it allows for the forearm to move. All right, so then to kind of sum up this entire video, if you consider eukaryotic cells, well, actually prokaryotic cells too, if you consider all the cells out there, it turns out that many of the parts inside of that cell work together in order to do things. Some of those things do some specific functions, like breaking down things and causing muscle contraction that's really important to the organism.